right, so we're going to talk about molecular modeling, and in particular, I'm going to start by showing you where on the internet you can find some 3D molecule files, okay, so that you can download them, open them up in various molecular modeling softwares, and look at them, manipulate them, etc. Okay, so probably the best place to look, the largest database that exists is called the PDB database or the Protein Data Bank. Okay, so if we search for that, we're going to see the top hit here is the PDB homepage. Okay, there is a wiki article on it, which you might want to have a look at. Basically, the Protein Data Bank is a database for the three-dimensional structural data of large biological molecules, such as proteins and nucleic acids. These are typically obtained through X-ray crystallography, NMR spectroscopy, or increasingly cryo-electron microscopy. Okay, and then after the scientists do the experiments and write up a paper or journal article, they would then submit their data to the PDB website, where it is freely and openly available for you and I and other people around the world to download it and look at the structures. Okay, so I'll click on the PDB homepage here, and there are a couple of things I want to draw your attention to here. One is the MOTM, or the molecule of the month. And this is a really cool program that PDB does where they highlight a different molecule every month. They've been doing this for years, and when you click on these, you'll see that it shows more about the structure, the history of those molecules, what they do. It's really cool. So you might just want to have a look through those, and you'll understand more about proteins and nucleic acids, how they work, what they look like, etc. So this is really cool. You should check this out. Second is a PDB 101. In this case, how do enzymes work? There's other PDB 101 courses which are also really cool. The main reason for coming here is to actually find some proteins or nucleic acid structures and download them. So I'm going to search for one of the most classic proteins. It is the green fluorescent protein. And click go. And we can see here the first one dates back to 1996. So this might be one of the first crystal structures of fluorescent proteins, actually. So 1996, Nature Biotechnology. It was officially released in 97. So yeah, this is cool. This could be one of the very first, because most fluorescent proteins are actually derived from the original green fluorescent protein. So this is cool. There are a couple of things here that we can quickly talk about. It gives you a highlight of how many residues or how many amino acids, so 476 in this case. The resolution. So the resolution is kind of the limit in which you can discern different atoms. Okay, so typically the resolution for crystal structures is between 1 and 2.5, we'll say, angstroms. And so this little a here stands for angstroms. The angstrom is 10 times smaller than a nanometer. Okay, so in other words, there are 10 angstroms in a nanometer. Okay, and remember that a nanometer is 10 to the minus 9 meters. All right. The reason angstrom is still used is that it is a really good unit for at the scale of atoms and bonds. All right. A nanometer is a little bit too big to really be working at that. Usually you're like 0.2 nanometers, which is kind of a silly number, whereas 1 or 2 angstroms is kind of a well rounded, or like a pretty good number, a whole number in a way when working at the atoms level of the atoms. Okay, so I'm going to click on this, and let's see, shows a little bit more here. It tells us X-ray diffraction is how they obtained the structure. There's a link to the more information on the publication, for example, so we can see the authors. And if we do click on this here, it'll open up a new tab, but also it tells us the organism it's from. It was actually expressed in E. coli, okay, so lab strain of E. coli. That's pretty cool, and it classifies it as a fluorescent protein. Right? I'm just going to click on this other tab here. So we can see there's a primary publication link here, so I can click on that, and it should take us to Nature Biotech 1996, and we see that the molecular structure of green fluorescent protein. So this is pretty cool. This is the original description. Unfortunately, it's behind a paywall. But yeah, so if you have access to this paper, you might be able to find it around the internet. You could actually see the original description that the scientists made upon completing the crystal structure. Okay. So the main thing here again was to find a nucleic acid or protein structure and actually download the file so that we can view it, manipulate it in some molecular modeling software. So I'm going to click on download files here, and there's many different options, but the most common file, which you can definitely start with, is the PDB format. Pretty much all molecular modeling software can read and open the PDB format. So if I click on that, it's going to download that file so that I can open it in the software. Okay, so we recently downloaded a file from the PDB, or Protein Data Bank website, for green fluorescent protein. And we are now going to open up this file and learn a little bit more about this particular piece of software called PyMOL. Now, PyMOL is one of the original molecular modeling suites, and it was originally open source and developed for many, many years. And then it was eventually acquired by a company called Schrödinger, which still keeps a very old version open source, but it's now closed source, and you need to pay a fee, a subscription, in order to use the newer version, which does have a lot of cool bells and whistles. And so as you go further into your molecular modeling endeavors, you might want to consider getting an individual license so that you can do some more advanced things. Okay, so again, PyMOL. So what I'm going to do is I'm just going to go up to my top bar here. I'm going to click on File, and I'm going to just look here for a moment. Okay, and so really there are two ways to open modeling files. Okay, so one is through Open, which you're probably very familiar with. You're just opening a file from your computer that you've downloaded, which we just did. But the second is to get it directly from PDB. All right, so I'm going to start by clicking on this one here, and it opens up this dialog where you can actually put in a PDB ID. All right, now what is that? So I previously had opened the file just to keep the code here, but when we originally were looking on the PDB website and we were looking at the green fluorescent protein, you may have noticed that there was a 4-letter alphanumeric code that was also kind of next to that molecule's description. And so PDB, they provide, or whenever a molecule is submitted, they assign a 4-letter alphanumeric code that is permanently attached to that molecule. And this is so that it's kind of like a barcode where any software can, if you put in that code, it will open up that particular file. Okay, and so I'm just going to accidentally close that. So I'm going to click on it again. So if we go ahead and put in 1GFL and we download it, there we go. It's going to open up that particular file. And so you can see now I've got green fluorescent protein visible in the window here. Okay, and I'm not even going to go through the other open feature here. You click on open, you find the file that you've downloaded, and you open it, and you're going to get to the same point that we have gotten to here. Okay, so that's a general opening. Maybe I'll talk a little bit about manipulating and moving around for viewing. So one important thing with PyMOL and other molecular modeling suites is that the way in which you navigate and manipulate the molecules depends on the computer setup that you're using. So, for example, I'm using a Mac laptop. I'm using the trackpad. So I will be clicking and scrolling around on the trackpad in a particular way, which might be different than you if you're using a mouse, for example, and with a PC or just a mouse in general. Okay, so if I go to the top of my program here and I click on mouse, then what you're going to see here is there's actually many different options where um, the creator of PyMOL decided, oh, okay, we can optimize your experience depending on what your computer is and what mouse you have, right? And so, for example, you can see here if you want to be moving the molecules around, and you have a 3-button mouse, meaning you have the 2 right and left clickers, but then you also have the middle wheel that is also a button. You might want to click on this and use that, or if you're editing the molecule, which we'll be doing in future videos, you might want to click on this, or if you're viewing with a 3-button mouse, you might want to click on this, okay? So really kind of have to just, you'll see some of what I'm doing here, and you'll just have to click around and learn the system based on what your computing layout is, okay? So I'm not going to click on anything. I'm just used to opening it and clicking around with stuff, okay? So for me, if I just click on my trackpad, which would be the equivalent of, I guess, a left click, and then I'm just moving another finger around the trackpad, I can rotate the molecules around in 3D, okay? So that's a pretty simple manipulation. So you might just need to left click and move your mouse around, and it might just swirl them around, okay? For zooming in and zooming out, I use the classic pinch in, pinch out that you might be used to using on a tablet or a mobile phone. So if I pinch out, I can zoom in. If I pinch in, I zoom out, okay? And if I zoom way out, I can still do the click and move the molecules around, okay? As you can see here. So I'll zoom in a little bit more. On my computer, if I want to move these molecules like left and right in space, I hold Option and then I can just click and drag back and forth. So that's how I do it. If you're using a 3-button mouse, you might simply be able to, let's say, click the middle button and move it around. But again, you'll just kind of need to play around with your setup and figure out how it all works. Okay. Other than that, that's pretty much how you can zoom in, zoom out, move the molecules around. Maybe I'll quickly just describe what we're seeing here. So we're going to do a lot more manipulations and viewings in another video, but I just want to describe that really this is a protein here. This is a green fluorescent protein. This one is a green fluorescent protein as well. You can see that they are identical for the most part. They're just, there's 2 of them and they're in slightly different orientations. Okay, and what we're seeing here is called a cartoon drawing, where we're not really seeing the amino acids that make up these proteins. They're a bit hidden, but what we're seeing is a computer-generated simplified drawing of where the backbone of the protein is, or the amino acid sequence, all right? Because as you'll see in later videos, it looks pretty crazy and confusing when you show all of the atoms, okay? So this is just, again, a very simplified view, which more or less shows us secondary structures. So for example, you can see there's a little alpha helix up here, here as well. You can see that there are these lines here with arrows, okay? And so these suggest a beta sheet, okay? And so you can see they're anti-parallel. You've got an arrow going up to the right here, and then down to the left, and then up to the right, and down to the left, and so on and so forth. And so that's an anti-parallel beta sheet. And overall, this structure is called a beta barrel. Okay, it's a very cool structure. It's quite stable, and there are a number of fluorescent proteins that have the structure. Okay, so that's why we're seeing this cartoon structures. It's really to help us get a basic understanding of the secondary structure of these proteins. Okay. And then the last thing I'll mention is we're also seeing these little tiny pluses here, or these little stars, right? And those represent solvent molecules. Okay, so recall that a solvent is something that you dissolve something else into, okay, or you dissolve the solute into a solvent. Okay, if you're making juice from juice crystals, you'll be dissolving the crystals, which is the solute, into water, which is the solvent, right? And similarly, as we learned a lot about cells and the operating environment of cells in the first Zero to Hero programming book on bacteria, um, proteins and in the cells, there's water everywhere, and that water is critical in allowing the biomolecules to complete the four Bs, right? Bump, bind, burst, and bump, right? There are these water molecules that are zipping around, solvating, binding to the outside of these proteins, for example, so that they can move around and interact with different things, okay? And so that's what these little stars are. They're solvent, and they're most likely water molecules that that remained in the crystal structure. They could also be salt molecules, but we can investigate that further a little bit later. All right, so in the last video we talked about opening a file that encodes for a three-dimensional biomolecule from PDB, and we showed how you can open it directly from PyMOL, or you can visit the PDB website, download that file, and then open it from your computer. And so in this video, I'm going to more or less go through how to select things within the loaded files that you have, because selecting the different molecules is really important if you want to manipulate certain parts of the system. All right, and so we have the same file, the green fluorescent protein file, open here. It's a slightly different color because it automatically assigns a different color when you open it. So the color really doesn't matter. You can change that, as we'll see in future videos. So we can see the proteins here. There's two GFP proteins, and then we have the solvents in the background. All right, so as mentioned before, the manipulating and selecting of things within your file in the open window here really depends on your computer setup. So again, I'm using a Mac laptop, and I'm using the trackpad. So the way I control the screen here might be different from how you control it with your computer, whether it might be a PC or a Chromebook, or maybe you have a 3-button mouse or a 2-button mouse. It really depends. So again, you kind of have to just play around and get to learn the setup with your computer. But I'll kind of take you through things. So one of the first things I want to mention here is you can either go to the top of the screen and click on mouse. You'll see a number of different settings that you can choose as you go through using PyMOL. I'm not going to touch anything right now, but you can, again, just with your system, click on different settings, and maybe you'll prefer one setting over another. Similarly, if you look to the bottom right of the screen here, this shows you your current settings with regard to moving things around and selecting things. And so you can actually click through. If I click directly on the 3-button viewing, I can actually change it. All right, so 3-button viewing, 3-button editing, so you can change that. And you'll notice some idea of if you're using let's say, your left mouse button and you're holding shift, then you can actually drag a box over top of what you're trying to select. Another thing you'll notice is what you're selecting could be molecules. I can click on that as well. I can click on many different things here. All right. So if I'm on 3-button viewing and I am selecting molecules, I can simply just, I'm going to do one click and it's going to highlight that entire molecule. You can see I'll do the same thing with the other. And in order to deselect, I simply click again. All right. If I zoom in and we look at these solvent molecules here, I can click on one of those and you'll see that I've selected molecule 376. Its composition is HOH, so that is two H's and an O, H2O, so that's water. So all of the solvents are water. I can again click on it and it will deselect, right? If I change the selecting down here to, let's say, residues, which basically means amino acids, if I click in the protein here, you'll see I only selected a particular amino acid, which we don't fully see right now because the atoms are hidden and we're only seeing the cartoon structures. But you'll see here I clicked on amino acid 163, which happens to be a valine. All right. And again, I can click on that and it will deselect. Right? The same thing you can do with chains. So a chain is also, these are two different chains. They're two different molecules, but they're also two different chains. All right. If you have a protein that has multiple domains or multiple molecules, they're also called chains. So that works the same. And you can play around with these different settings. All right. So that's the basics of selecting and deselecting. If I go to, let's go to atoms, and you'll see here that in this current setting, if I left click while holding shift, or if I hold shift and I left click, let's see what that does. So it creates a box. And when I finish clicking, it'll actually select all of the atoms within that box because I have the atoms selected down on the bottom left here. All right. And if I just click anywhere else, it will also deselect all of those atoms. All right. So that's the basic manipulation with selecting and deselecting. And as you'll see, if you want to let's say, you can, let's say we zoom in here. We're clicked on atoms right now, or let's click on molecules. As you'll see, if you click on that, you'll be able to delete that specific molecule later on. Okay, and we'll get to that. One other thing to mention is the right clicking. So if I double tap on my Mac, which is the equivalent of a right click, it's going to pop up a dialog box here which has many different options. Okay, so I can inspect, I can do certain actions on what I've right-clicked. Okay, so for example, what I've right-clicked on, I could remove the atoms if I've selected it. I can do many different things, right? So if I click on that molecule and right-click, I can do many actions here, which we'll get to later because this is sort of another version of what we'll explore over here. I just want to bring your attention to the idea that you can right-click on the molecule and that will present you with options on how you might want to act upon that molecule. Okay? Okay, so how can you act upon molecules within PyMOL? All right, so we just talked about the right-clicking, but now I want to draw your attention to the top right part of the screen here. And you'll notice that there's currently two levels. I'm just going to click on something here and click off of it. You'll see that a new level appeared. And so there's really two ways to look at this section. There's vertically, so notice that there's three different lines here, but then also horizontally, where you have a number of little boxes associated with each line. Okay, and so really there's three levels of how and what you are going to change within this program. All right, so the top tier here which says all, basically means that if you use these little boxes here, which we'll go into in a minute, on the all line, it's going to apply those actions or changes to all of the loaded structures and atoms and molecules. Okay. Underneath, you'll notice the 1GFL, which we know is the 4-letter barcode for the fluorescent protein. If we use these boxes on this level, it is going to apply to all of the atoms and molecules that were loaded as part of this file. Okay, and you can actually load multiples of different PDB files. So if you had multiple loaded, you would actually see the 4-letter designations all the way down. Okay, and then we have SELE, which stands for selection. All right, and we just went through a quick video on selecting different atoms or molecules. And so if you select something and then use these little boxes, when you click on it, it will only apply to what you've selected. Okay, so let's go through that now. The first box here, A, stands for action. And action is something that you can use to affect change in the molecule. All right, so as you'll see with the other boxes, they more or less will help you change what it looks like, what's visible or not, and maybe colors. But they don't actually change the physical makeup or the atoms or bonds within the system. Action can change what's there. You can delete atoms, you can remove waters, you can add hydrogens, and you can compute things and more. Okay, so action is one of the key things you'll use later on to actually change your model. Okay. And again, if you use the action at the all level, it's going to modify everything within the system. If you use it at the selection level and you've selected something, it will only modify what you've selected, right? So if you select some water molecules and then you click remove atoms, it's only going to remove those selected atoms in the water molecules, okay? Next over we have S, which stands for show. Now show is simply something that will make things visible that you don't currently see. All right, so as we talked about earlier, when you open files, it'll typically default to only showing the cartoon structure. Okay, and remember the cartoon structure is a computer-generated model of maybe where the backbone is of the amino acid structure, or if it's a nucleic acid of the nucleic acid. And it doesn't really show you any actual atoms or molecules or amino acids within that system, all right? And you can use the show tool here to actually show atoms and molecules. If PyMOL loaded your proteins or nucleic acids showing every atom, as you'll see later, it's pretty confusing, and it's really hard for us as humans to see patterns. And so by showing just the secondary structure off the get-go, it really sets a baseline of understanding what we're dealing with. Okay, so if I, on the all tab here, if I were to go down to, let's say, main chain, which refers to the amino acid backbone, and I click on sticks, it's going to show the backbone, the amino acid backbone, throughout those proteins. Okay. If I select on one of these and I then go the selection and I show, let's say I want to show the side chains now as lines. So this is just a thinner visual than the sticks. There we go. So now I've only shown the side chains of the amino acids on this right side here. Okay. And then H stands for hide. So that's the opposite. If you want to hide certain things, to view differently or inspect things differently, that's also useful. All right. So, for example, if I were to select the left GFP protein here and I'm going to hide something in that selection. Let's say I want to hide the cartoon, which is basically the secondary structure. If I click on that and click away, now I can see the backbone alone. Okay, and because we saw the cartoon earlier, we can sort of see the beta barrel structure, although it's definitely more difficult without having the cartoon representation there to help us along. Okay, and you can hide lots of different things. You can hide waters or hydrogens, lots of different things. You can click on the show and the hide to learn what all of these different things are. One that's pretty cool, I'm going to select on the left here. I'm going to go back to the show. You can actually compute the surface of that protein. Okay, so I'm going to deselect now. And imagine all of the amino acid side chains and everything. Imagine the van der Waals spheres where it's kind of like the edge of each one of the atoms. This would sort of be the surface of the protein. Okay, so that's a pretty neat one. I can let's say so it's harder to see whether I've selected this or not. Let's see here. I'm gonna click off. I'm gonna click on here. It's harder to see when you have this surface on. You can't really see the highlighted atoms underneath. But let's see if I have it selected. I'm going to hide the surface. No. Let's try now again. I'm going to hide the surface. For whatever reason, this isn't working. So, okay, what I'll do now is, because I can't see the selection right now, I'm actually going to go to the this area here, and I'm now going to hide the surface, right? So now I don't need to select anything. It'll simply hide any surfaces within that loaded molecule, all right? Another one that is interesting is, so similar, a mesh is like a surface. So I'm going to go show mesh, and it's just like the surface except you can see through it, right? So it just is a mesh on the surface. It's a pretty neat one. I'll now go and hide that mesh. Okay. If I select one of them and let's show spheres. So again, the spheres are simply an estimated outside border of the atoms, right? So there's the electron orbitals, which are around the nucleus, and those orbitals go out a certain distance, and these spheres represent kind of how far out that electron cloud will go, and represents kind of the outside of those atoms, right? Because if you have two atoms coming together, they won't. Those spheres will act as kind of like hard balls, and they'll hit one another, and they'll repulse because they're negatively charged, and you won't see them overlapping, right? The only time you see them overlapping, like in this benzene ring here, is if there's covalent bonds, right? So that means there's been enough energy added to the system that actually allows two atoms to come together and share electrons, and then you see the overlapping, right? But that's within a molecule. If you're talking about side chains of different amino acids, they're not just going to come together and overlap, right? Because they're discrete systems, right? And so you won't really see any of these hard balls overlapping with different side chains, okay? So I'll hide that. Spheres. And then the last one here, second last one, L stands for label. So you might want to label atoms or residues, things like that. And so if I just click on here, I'll label the selected. Let's go with the residues, and it's going to label every amino acid that's within this selected region, right? So you can see up here we've got lysine, glutamate, asparagine, proline, arginine, so on and so forth. And that can often help you to know what you're dealing with, especially if you're engineering the protein. You often want to know where the different amino acids are that you're maybe wanting to manipulate or change. Okay, so I'm now going to, you can't hide the labels under labels, you actually have to go to hide. So I'm just going to go hide labels, okay? And then the last one is C, which stands for color, right? And there's many different ways you can color. If I select this one on the left here and I use the box under the selection for color, let's say I just want to go red, fire brick red, and I deselect, it's going to change all of the atoms and structures that are visible. It's going to make them red, okay? Now, one thing which is kind of important in chemistry is that there are color designations that are standardized for the different atoms, okay? And let's go here by element. So you can see you can actually color by element. And the international rules or standard around coloring atoms is that carbon is generally a charcoal or a black, hydrogen is white, nitrogen is blue, oxygen is red, and sulfur is typically yellow. All right, and you can see in here these aren't really adhered to, and the closest, I guess, is maybe the top one here, except the carbon, for whatever reason, isn't introduced, okay? And so if we, in order to have the official coloring, one thing we have to do is we actually have to change the whole molecule first to like a gray. All right, so now everything is gray, but then if we go back and we go by element and we click on this top one, it will then change all the oxygens red, all of the nitrogens blue, and then the carbons will all remain gray. All right, so that's pretty cool. And we can't really see the hydrogens here, but let's show those. So if we go click on this, and then again, we're working on the selection area here. If I go action, hydrogens, and I want to add the hydrogens, now you can see them all visible, all right? So all of the hydrogens on the backbone are now visible, and they are white, which follows the international standard, all right? So those are the general tools for manipulating and showing different things within proteins or nucleic acids, and that can really help you to learn more about what your system is and distances between things, and just to get a general idea of what you're working with. In the future videos, we're going to be covering more manipulations and more analysis. In this video, we're going to go a little bit deeper into analyzing these protein structures as well as doing our first mutation. So in general, one of the best things to do is to simplify your model if you can. And so as an example, I'm going to click on the all, and I'm going to remove the waters because I don't really care about those now, so I've removed them. I'm also going to remove one of these two GFP molecules. So I've clicked on the select, I've selected one of the chains. If I go to the selection here, I'm just going to go to remove atoms, and then that's going to remove all the atoms. And I'll slide this to the middle, and now I just have one protein loaded, so it's a little bit simplified, okay? So the first thing we'll do is let's do some measurements to figure out the height and maybe the width of this protein. And the way we'll do that first is let's show the side chains of this protein. So I'm just going to go to the S for show. I'm going to go down to the side chains, and I'm going to do sticks. All right, so now you can see all of the side chains here. All right, and if we look at the top and the bottom, it's pretty much, we've got this amino acid side chain, you could argue is the highest, this one is the lowest. And so let's see what the height of this protein is, and then it looks like this amino acid is the most to the left and this one most to the right. And so we'll use those to get a better understanding of the height and the width of this protein. All right, so what I'm going to do is I'm going to go to the top, I'm going to click on Wizard, and then if you look down, there's something called Measurement. All right, and so now it'll ask you here, please click on the first atom. So I'm just going to go ahead and click up here, and then I'm going to go all the way to the bottom because it's asking me to click on the second atom, and there we go. Okay, so now you can see that there's this yellow dotted line that goes from the top to the bottom, and you'll also notice that on the side here, measurement 1 came up. And this is so that we can change the color later and change more or less what it looks like. Let's go ahead and do the same thing with the left and the right here. So I'll just click on this atom here and this atom here. All right, and now we've got another measurement that comes up. And I'm just going to click on the bottom right here, done, because if we keep going, if we don't click done here, if we click at all in this model, it'll count that as the first atom and second atom. And so I'll just click done here so we can look at these a little closer. If we rotate this molecule, we can see the width of this protein is 35.9 angstroms. So that's 3.59 nanometers. All right, and then if we rotate it so we can see the height, we've got 52.3. Zoom in here. 52.3 is the height in angstroms, so it's just over 5 nanometers tall. All right, and if we wanted to, we could click on these different measurements. So measurement 2 is the width, measurement 1 is the height. So let's say I want to color one of them here magenta. There we go. So now it's magenta. We might be able to change that label. No, it won't let us click on the label, so we can't change that. We can hide dashes and we can hide the labels. So you're a little more limited in what you can do here. But you can still modify these a little bit. All right. We can also figure out the distance between maybe just two residues here. So if I were to zoom in here, let's drag this down. Let's drag this down here. Let's just look at these two side chains here. Maybe we could just do a measurement to see how far apart they are. So I'm going to again go back up to Wizard, Measurement. And again, it's asking me for the first atom. I'll just click there. Second atom here. And we can see we're looking at, and I'll click done here, we're looking at 6.9 angstroms between these two, okay? So that'll be measurement 3. I'll change the color of that one to cyan, aquamarine. And I wonder if we can change the label. Can't change the size of the label. No matter. So there you go. That's how you do measurements. And you can learn a lot about the system, the spacing between amino acids, side chains, or really any atom, the height and the width of the system. And you can even measure between like the active center of the protein. So for example, in the green fluorescent protein, this inside part here where you're seeing this alpha helix, that's where most of the magic happens, where the light is absorbed and it fluoresces a particular color. So you could even click on some atoms inside there and see how far they are from the outside of the protein, for example. So there's a lot that you can learn by simply looking at the distances between things within a protein. Right, so in this video we're going to manipulate the proteins. We're going to do site-directed mutagenesis, which is when you choose a site and you mutate it. So for example, we can choose an amino acid and we can change that to another amino acid. All right, so I'm starting out at the beginning, and again, we're going to simplify this a little bit. Okay, so one of the first things I'm going to do is remove the solvent. So if I click on all and then go down to remove waters, that's going to do that. Another really useful feature is to right-click on a particular part of the model, and you can choose, like I right-clicked on the right-hand GFP molecule. Now, technically it's considered a chain, and there's two chains in this. Each of the GFPs is a chain, and so I can select this chain, and it's going to highlight over here. And then when it's selected, I can actually remove the atoms. So that's one way of deleting one of the chains. Okay. And so now it's a lot more simplified here. I can show, I'm going to go ahead and show side chain as sticks again. All right. And let's mutate one of these amino acids. All right. Let's keep it simple. Let's maybe focus on an amino acid out on the peripheral. There we go. Let's maybe choose this one here. All right. This amino acid, which is a GLU 172. All right. So that's a glutamate amino acid, and it's amino acid 172. So actually, one thing I'm going to do now is also show you one more feature here. If you go to, let's find it here, display and sequence. If you click on sequence, it's going to show you the amino acid sequence at the top, and you can use this scroll bar to go down, and you can actually see that wherever there's a little green highlight here, that's where something has been selected. All right, and so I must have accidentally selected an amino acid down here. Actually, yeah, you can see I selected an amino acid down here just by clicking earlier, and that comes up here. So you can also just click on them on the top, and they'll either be selected or not. You can actually click and drag, and select entire chains. It's very useful. You can see them being selected underneath, right? It's kind of neat. And it's very useful. You can use this with nucleic acids as well. All right, so I'm going to zoom back in here, and we're going to use, if you go to Wizard, and then mutagenesis, and then protein. All right. And so you'll see again, there's a cue here which says pick a residue. And so I'm going to click on this glutamate residue here, okay? And you're going to see this gray other version kind of pop up there, okay? So it's also glutamate. And if we look to the right here, there's the little mutagenesis box that's open, okay? And so currently, there's no mutation, and that's why we're still seeing a glutamate residue here. But if I click on this box, I can actually choose some of the different amino acids, okay? And so, for example, I could choose isoleucine here. If I choose isoleucine, you're going to see it actually changes it. It's still gray because you haven't clicked apply or done yet. So you're still seeing the original, but it kind of overlays the other amino acid you want. So if we want to choose a different one, like asparagine, right? Asparagine overlays a little bit, but it still shows you the original. Okay. We can add and retain hydrogens. So for example, if you're adding a new amino acid in and you want to see all of the hydrogens, you can click on this and you'll see now all of a sudden we can see all of the hydrogens. Okay? Because remember, in chemistry, often hydrogens are not shown even though they're there for simplifying the models. But in this case, if you're doing mutagenesis, you might want to see where all of the atoms are, including the hydrogens, so that you can see how they might clash with neighboring amino acids or other atoms. You can have, so rotamers, meaning you're basically rotating the backbone here. You can be independent of the backbone, or you can be dependent of the backbone, and then you can have caps on your amino acids, right? And so because we're in line, we're actually in the protein, we don't really want to put a specific cap on the either N, the amino end, or the carboxyl end, because that's going to terminate and separate it from the amino acid chain. So we're just going to leave this open. It's going to fit into the chain here. Okay. All right, so if that's good for us, if we want to change this to an asparagine amino acid, we can click apply, and then all of a sudden we now have a mutated asparagine in this protein. Right? And so that's the first step to mutagenesis. In general, you then need to do what's called sculpting. You need to see how that impacts the overall structure. And I'm going to click done here. All right, and so now that's a permanent part of the molecule here. Right, so if we click on it, we can see this used to be a glutamate. Now it's an asparagine. Right, and what we can now do is, if you go into Wizard again, you'll see there's something called sculpting. And sculpting is basically means it's going to try and energy minimize the structure so that it mimics what it could actually be like in real life, right? Because we just kind of put this this amino acid in, it may not fit nicely in its environment. There might be bonds or charges in its proximity that exist from other amino acids or solvents. And when you just popped it in there using a mutagenesis tool, you didn't really consider any of those other environmental factors. And so sculpting is a way to kind of understand whether your model is maybe a little more real. And so in this case, I just clicked on sculpting. It asked me to choose an amino acid as the central core, and I clicked on ours, and it you noticed it moved a little bit, right? And so technically, that would be some energy minimization. And so you can see here, for example, you've got a hydrogen coming off of the backbone here, and it actually moved so that it's kind of pointing at this oxygen. And my bet is there's a polar contact there, or a hydrogen bond that is forming between those right there. All right, and that's basically it. It's if you solvate this protein or add waters, then you'll have an even more real idea of what's going on, because this amino acid will most definitely contact water or solvent molecules in the environment because it's on the outside of the protein, right? Whereas if you were to choose to mutate a amino acid that's inside of the protein, then it's not necessarily going to be influenced by solvent because it'll be embedded in the protein, and so solvent may not matter in that case. But in this case, it's pretty cool that you click on the atom, it isolates it from the rest of the protein, and then does the energy minimization. And this is pretty important because to do energy minimization on this entire protein would take a long time and require a decent computer. But by isolating it to a region where the majority of the impacts are involved, it helps you to do a rough model right off the get-go. All right, and so it looks like we can click on here though in the sculpting. So we've isolated it. We can create a solvent shell around. Let's see here, if we click shell and then the radius of 5. Oh, this is basically how we isolate the area around that we're going to be doing the energy minimization. So if we do the radius of, let's say, 10 nanometers, it's going to be slightly larger. And yeah, so we can click done, and now we've got our amino acid in there, and it has been energy minimized so that it's somewhat like it would be in nature. Okay, so in this video, we're going to look at how we can mutate inside the protein. In the prior video, we changed an amino acid on the surface of the protein, and this time we're going to go inside of the protein. So similar to before, let's simplify this a little bit. So we're going to remove the waters here. I'm going to right-click on one of these and go down to chain and just go to remove atoms. And now we have one of the proteins here. So this will simplify things for us as well as make sure the computer can handle some of the modeling that's going to happen. Okay, so now what I'm going to do is I'm going to show the side chains. I'm going to show sticks again so we can see where all of the amino acids are, but I'm going to keep the secondary structure. I'm going to keep the ribbons so we can see the general structure of the protein. And let's have a look here. So if we look inside, so for example this alpha helix here, we can see that there's an amino acid here. This one here, for example, we've got a little valine. Right, so maybe we can change the valine to something way different just to see what happens. And so valine is very hydrophobic. And so perhaps if we threw a charged amino acid in here, it's going to really change the structure. Okay, so let's try and do that. Okay, so let's just move it so I can see this area quite well. All right, so similar to before, we're going to click on Wizard, and we're going to go to Mutagenesis, and we're going to choose Protein. And it's asking us to pick a residue, so I'll click on it, and that'll deselect, but then I'm going to click again so that it knows that's what I want to change. And you can see, look at all these clashes that are happening here. We've got this giant amino acid now that's very close to some of these other amino acids. So there's definitely going to be a clash here, right? There's going to be a clash here. So we'll see what happens when we start sculpting it. Because you can see this is a glutamate, okay? So we'll keep it at this one here. So let's go ahead and I'm going to click Done. Let's try that once more here. Wizard, Mutagenesis, Protein. I'm going to choose this little valine here. We need to click Apply, and then Done. All right. Okay, so now we've got this glutamate that's in here, and you can see we've got, this is going to be a negatively charged oxygen here. We've got an oxygen here. So there's going to be some clashing happening when we do the sculpting now. So I'm going to zoom out a little bit. We're going to keep an eye on it. In fact, I'm going to click on here. I'm going to change the color so you can see it even better. Let's change it to hot pink so you can see it. All right. Deselect it here. And now we're going to go Wizard, Sculpting, and similar to before, it's going to ask us to choose the center atom, all right, of this amino acid. But let's set these here. Let's constrain it to, let's say, 6 and a cushion of 6 as well. And then let's click on here, and you're going to see it, things changing and moving immediately. So keep an eye out for that. So you can see how it immediately twisted here, and now we have, things are still moving. So this ring, for example, is slowly twisting. The fan on my computer just came on, so the computer is doing a lot of computations right now to try and figure out how to make this fit. And because it's constrained to a particular area here, that can actually limit the model from finding a good solution, because currently all of the gray amino acids are not going to be moving. They're excluded from the model. And so it's possible that by putting this charged amino acid in the center, all of the other amino acids can't shimmy around and move around enough to account for putting this charged bulky amino acid in there. And so it might be required to actually increase the overall radius to much larger, and then the cushion in which we're going to kind of constrain the energy minimization to, if we increase that as well, we're actually going to start to see amino acids much further out moving as well. It looks like most of the movement is complete. So if we look in here now, we can see that whereas before there were lots of things that were quite close together, they're now pretty far apart. So for example, we've got the oxygen here and here are pretty far apart, same here. So there's a lot more spacing in the center of this protein now. Now, by putting this inside and doing the sculpting, we're able to see what a structure could look like after it's being minimized, but this doesn't really tell us much about what it's going to do to the protein within the cell. So for example, will this protein still be fluorescent or exhibit fluorescence? Will it change the color of the fluorescence? Will the protein even fold properly? We don't really know from this. All we can really tell is whether this mutation can even kind of fit within the protein. And we can see that all the other amino acids were able to move away from it and create some space. There was no major clash happening. Sometimes if you do energy minimizations, when you mutate, you actually can see different amino acids like combining to form a structure that would never happen. And then you know that there's no way that mutation can actually happen in that constrained environment. So in this case, I'm going to click done. I'll click on that amino acid. Again, let's turn it hot pink. And in this case, we could then see where our mutation is within our protein, and then we can log that. We could even then save this file. If we go File and Save Session, you can either save the session so that it'll open up the environment just as it is now, or you can export the molecule so that you create your own new PDB file. Okay. Or you can even export an image. So for example, you can create a PNG. Now ray tracing here, so you can either show this exact image that you're seeing, or you can do what is called ray tracing. And this is where the computer will simulate this being in a room and it'll shoot light at it, and then the light will bounce around just like in real life, and it'll look maybe a little more more cool. So let's go opaque background. Let's save this crystal structures as mutated GFP. In this case, often the way you label mutated proteins is, so you can see up here, this is amino acid 61, all right? So what you would typically do is you do it like this. So we started out with a valine. The amino acid, one-letter code for valine is V, and then you can say 61, 61st amino acid was then changed to a glutamate E. So in this case, you would have your GFP, the mutation is valine number 61 was swapped to a glutamate residue. Okay. So I'm going to click Save. It's going to create an image. You can see there's a lot more light on it now, and it's not a great image because it kind of hides our amino acid a little bit, but you can see the sunlight or the light source and the shadows formed by these different things here. And you can see with this open version, it actually puts a watermark on it. Okay. There we go. So that's how you can mutate. And actually, one exercise you can do is you can do measurements. So in a previous video, you could measure between two amino acids or two atoms. And then what you can do is you can do the mutation and then measure again. And then you can even do the sculpting and then measure again so that you can get a good idea of how the atoms have moved and the spacing between them. Okay, in this video we're going to build a peptide chain. All right, so in most scenarios, when you're engineering proteins, you will be mutating a number of amino acids to something else, or you might be adding a short segment, a peptide, into your protein, or you might be linking two different proteins together. And so a key part of that is to know how to build a short peptide sequence so that it can be either linked to other proteins or added into a protein. And so that's what this video is about. And so if you go to the top build link here, you'll see drop down and you'll be able to build based on different smaller precursors. And so in this case, we'll go to residue, which is just another word for amino acid. And we will be able to just choose the peptide we want to create. So an example might be, let's start with glycine. Glycine is a small amino acid which doesn't have a big bulky side chain, and that means it can kind of fit into most places in proteins. It doesn't have a big impact, and so glycine is always a good amino acid if you've got a tight place to put stuff. It also means that the chain you're building will be a little more flexible because there isn't a big bulky side chain that's either charged or not charged that requires a certain amount of space, and the more atoms you have, the more bonds you can have, which means that it can be more rigid, for example. So we'll just do glycine. Heck, let's do another glycine. It's going to continue building, and you'll see this little tiny sphere here, and this little sphere indicates the central atom in which you're adding a new molecule onto, okay? And so in this case, you'll see here that this is the C terminus, and you remember when a cell or when a ribosome creates a protein, it always creates it from the N, the amino terminus, which is over here, in the C terminal direction, right? Because remember, the C terminal amino acid, the amino acid at the very end, always has a free carboxylate group on it, all right? And in this case, it's just kind of showing this terminal carbon. We don't see the other oxygen here because that is where the next amino acid is going to be added onto. So we don't see the full picture here yet, but basically this little sphere, which if you look on the right here is PK1, is where the next residue will be added. So if we did 2 glycines, let's continue building glycine, glycine. What about glycine, glycine, lysine? So it's going to add a big bulky lysine on there. You can see that previously we had this terminal carbon here. It connected to the amine group of the next amino acid, right? So now we have a direct carbon-nitrogen bond here. And then we've got, this is the side chain, all right? And then now we have another terminal carbon, and this is again, a bond is going to be created with the next amino acid, and this will end up being a carbon-nitrogen bond right here, okay? So let's build and add another residue here. Let's go with, I don't know, serine, why not? So now we've added a serine, so you can see again, this is the carbon that was at the C-terminal end, but it is now bound to the nitrogen of the new amino acid, right? And then again, the carbon, we can see the side chain here of this amino acid, and then again, we've got the C-terminal carbon right here, all right? And let's just add one more residue here, and just keep your eye on that C-terminal carbon as I click on tryptophan, it's a big amino acid. Boom, all right? So you can see that this is where the terminus was, again, where the amino acid chain extended through a carbon-nitrogen bond. And then now, again, we have the C-terminus, which is over here. But one thing you'll see is like, wow, these amino acids are very close to one another. So that's probably not real. We'll need to do some sculpting to this, okay? So now what I'll do is, just so we can keep an eye on things, well, I'm going to add labels. I'm going to label the residues so we can see them. I'm also going to remove, you can see this back, it's a cartoon in there. That's not supposed to be there. It's just to help us see what's going on. So I'm going to hide the cartoon. So now we just see the backbone, right? And what I'll do now is, because, so if we look at the N-terminus, for example, so a nitrogen atom generally has 3 bonds connected to it. Right now we only see 2 bonds, and that's because we're just not showing a hydrogen here, all right? And so what I'll do now is I'm going to go hydrogens, and I'm just going to remove the hydrogens just so we are at a 0 hydrogen state, and then I'm going to do the same hydrogen and I'm going to add them, okay? And now we're going to see hydrogens at all of the spots. So for example, at the N terminus here, you see a proper amine, right, at the N terminus. At the C terminus, it added a hydrogen, even though technically this would be an oxygen atom here, because again, it's a carboxyl group, but the computer was like, okay, there has to be another bond here. I guess it must be a hydrogen. But when we add this into, or combine it or link it later on, this is going to be, this is going to disappear, and a bond is going to be formed here with the amine of the next part of the chain. And similarly, one of these hydrogens is going to be gone, because that electron will need to bond to the C terminus of part of that other protein. Okay. All right. So now maybe let's go ahead and sculpt it, because when we do add this into our other protein, or tag it onto that protein, then it's nice to start with a structure that is somewhat realistic. And in this case, this is not realistic at all, especially here. So let's go ahead and do some sculpting. So we're going to go wizard and then sculpting. And I'm just going to click on the center of this, peptide chain here. Why not right on the lysine carbon here, and we're going to see that. So there was definite clashing happening here, and so they separated quite a lot. Still separating a little bit more. The tryptophan side chain is rotating out a little bit. Let's include the whole thing here. So you can see the backbone is shifting a little bit. All right, so it's quite a different shape than it was before. These spread apart. The backbone twisted a little bit here. All right, so I'm going to click Done. And so now we have our peptide that we're going to see if we can, well, we'll do a few different things with it. And so in order to hang on to this, I'm going to export the molecule. All right, and we're just going to leave the settings how they are. Why not? And we're going to save this as, we'll just call it peptide. Okay. All right, so this was now saved as peptide. I'm going to delete everything. In this video, I want to start out by, before we get into linking this to other proteins and whatnot, I want to start by talking a little bit more about like the sculpting feature, for example, right? So the computer has these algorithms which kind of guide the twisting and the turning and the movement of the atoms, but I want to talk a little bit more about that, just so we understand, for example, bond angles and whatnot in the backbone especially of amino acids and proteins. Okay, so this is the peptide we created. It is the cartoon structure right now. So I'm just going to go ahead and I'm going to show the main chain as sticks and the side chain as sticks. And I'm going to hide the cartoon. And we're sort of where we were before. Again, it's a bit of a different color because PyMOL just assigns, seems to be random coloring depending on when you're opening things. And so I want to draw your attention to the backbone here. And so we can see that there's slightly different twisting in this backbone, okay? And I want to investigate this a little bit more because as you get into engineering proteins and adding more amino acids in or creating linkers like this, it's important to note that this cannot be twisted in any way you want it to be twisted, okay? In nature, there are certain rules that you need to follow to make your model realistic, right? And the goal of molecular modeling is to create as realistic as a model as possible so that you can often create that in the real world through genetic engineering and hopefully have an informed and increase our chances of success in the real world. And so we're going to quickly talk about these bond angles here. And in particular, we're going to talk about the angles between the carbon and the nitrogen and the carbon and the alpha carbon here, okay? And in particular, they're called the psi angle. So in between the double bonded oxygen carbon and the alpha carbon, that's called the psi angle, and between the alpha carbon is the carbon that has the side chain coming off, and in this case it's just a hydrogen because it's glycine, but then that carbon and the nitrogen, that's called the phi angle, all right? And I'll quickly draw your attention to, we can search for something called the Ramachandran plot. And something will come up here. Let's look at Wikipedia. All right, so let's, what I was just talking about here. So if we look at this carbon with the double bonded oxygen, that is the psi angle. And then from the alpha carbon to the nitrogen is the phi angle, all right? And these are really important for the backbone, and there's only certain ways that these can twist. And in many cases, it's because if we look back at our structure here, imagine if you twist these backbones so that, let's say you have the oxygens pointing in the same direction, then you're going to have like negative charges on the same side, which can actually cause repulsion. And if you twist them, then you might also have hydrogens on the other side, which are slightly positively charged, which can cause repulsion, right? And so because of that, the amino acid chain is actually going to relax, and it can only be in a certain orientation, which is sort of governed by bonding and electrons, right? So let's go back to this wiki article here, and I'm going to go to, here's a really cool Ramachandran plot. Now, what this shows us, each one of these dots is a real example of a bond or different bonds that are found in nature that have different psi and phi angles, all right? And you can see that all the areas where there's a lot more dots means that these are more often found in nature, and the areas where there's no dots, basically it's not really seen in nature at all, okay? And again, it's because of the twisting and which causes, different, like the oxygens to kind of repulse one another, and so it just wouldn't normally happen in nature because of that, right? And so you can see here with reference to secondary structure, alpha helixes, for example, which is a very common secondary structure, the psi angle, so again, between the carbons in the backbone, often the psi angle is close to zero, plus or minus 60 degrees or so, and the phi angle is at about minus 90, we'll say, right? And then in beta sheets, it's a little bit different. The psi angle can be anywhere from, we'll say, 90 to 180 for the most part, and then the psi angle can be minus 90 to minus 180 sort of thing. And then there's some other structures over here which are also possible with different angles, okay? And so I just really want to drive the point across that, again, you can't just twist your peptide chain or your protein backbone any way you like because it just doesn't work in nature like that. You need to adhere to the rules of bonding, and it's really just bonding in electrons, okay? So let's do an example here where let's take the peptide that we have created, and let's change the psi and phi angles to 0 and 0. All right, so let's see what it would look like if we had made the angles sit right here where we clearly see no dots. Okay, so our hypothesis could be if the phi and the psi angles are 0 and 0, we will likely see some sort of clashing between atoms, causing repulsion or overlapping of van der Waals spheres, which suggests it wouldn't happen in nature. Okay, so that could be our hypothesis, and let's go and check that out. So I'll go back to PyMOL. All right, we're going to see our molecule that we have created. And one thing you'll have to do if you want to do this is, if you go down to the bottom right corner, I was previously on viewing in the past, and I'm going to have to click on 3-button editing, okay? Because now we're going to be editing the main chain here, and I need to click on atoms and joints in order to start messing around with these angles in the backbone, right? And so again, we have the psi angle, which is between this carbon and the alpha carbon here. And if I click on the middle, I'm going to hold control on my computer. That's kind of what I need to do. Then I'll be able to start moving and twisting. Okay, so I'm going to hold control, and you can see I clicked, and now it's showing me that the bond angle is normally minus 30.4, and I'm actually going to twist that so that it's 0. Okay, so now we've got this bond angle that's 0. And I'm going to do the same thing with the phi angle. And let's see what it is at. It's at it's at 175. So we need to make that 0 if we can. Oh, it doesn't really want to, does it? Should be able to make it. It's just a matter of getting, okay, there we go. All right, so now we got a phi and a psi angle that are both zeros, and look what we can see here. So I'm just gonna go back to viewing, slide this over. Look at this angle. We've got two oxygens, which are very negatively charged, which are now almost touching one another, all right? And so there is a lot of repulsion happening here. Without a doubt. And so I'm also going to, let's look at the van der Waals spheres of these oxygen atoms, okay? So again, the van der Waals sphere is kind of the outside of those atoms. That's kind of how far the electron orbitals will go. And I'm going to, on my 3-button viewing, I'm going to hold Shift, and I'm going to draw a box, except I want to go around the atoms. So I'm going to change this to atoms. I'm going to hold Shift, and I'm going to try and just get these oxygen atoms. All right, it looks like I got this carbon here, but I was able to unclick it. You can also just click on them if you like. And because I've selected those, I'm now going to show spheres. Whoa, okay, so now you can see these oxygen atoms are just, they're overlapping one another. And this wouldn't happen because remember, these electron orbitals, they're kind of like hard spheres, and the only time you see overlap is within a molecule, right? When you have a covalent bond between two atoms. And there's no covalent bond between these two atoms. So this is why at a 0 degree psi, 0 degree phi angle situation in the backbone, you would not see in nature because the repulsion here would be enormous. And so let's do something fun where let's do some sculpting. Let's zoom out a bit here and let's go again to Wizard, Sculpting. I'm just going to increase these to make sure we get pretty much the same, the whole molecule. And I'll go ahead and I'm just going to click on this nitrogen here. And let's watch it. I'm going to zoom in a bit. Let's watch this this change, okay? Look at that. It totally changed the angles. So again, these spheres, the outer edge of these oxygen atoms do not touch with one another, okay? And they do not overlap. So I'm going to stop this now. And you can also see that it looks like these moved even further apart, which is interesting. Okay, so these spheres are just touching now. They're no longer overlapping. And so the computer suggests that this is a possible state. Okay, there's no crazy overlapping happening between the different atoms. Now, this raises another really important point, and this is that when you're making a molecular model, the starting point in which you have your model and then you run computations on can affect the outcomes a lot. Okay, and this is because the computer, it just has a starting point and it's going to try and find like the first solution. And there can actually be lots of solutions. And let's just kind of discuss this a little more. So I'm going to go ahead and hide the spheres. All right, and let's look at what these bond angles are now and see where they reside in the Ramachandran plot. Okay, and so again, I'm going to go to 3-button editing. I'm on atoms and joints, and I'm just going to click, oops, with control here, minus 12 psi, minus 73 phi. So let's go minus 12. Okay, so we've got minus 12 psi. So maybe like here, and minus 73, so minus 90 is about here for phi. So minus 73 has got to be around here. And then we've got, again, minus about 12. So actually, it looks like the computer has optimized this molecule to kind of be in this area here, one of these dots. Let's just double check to make sure. So let's go back here. Again, we've got minus 73 phi. Oh no, we got plus 73 phi. Good thing we double checked. And minus 12 psi, which is in this region here. Okay, so this is about 90 here, so this is probably the 70s, and then minus 12-ish is going to be, here's 45, so about here. Okay, so when we look at this, it looks like the computer has optimized this backbone to actually be in this segment here. Right? So it's not as frequent as some of these other areas, but this is considered within the realm of possibility. And so then you have to ask the question of if you had twisted that backbone slightly differently, like let's say it wasn't at 0, 0, what if you went negative 20 psi and 0 phi, or sorry, minus 20 phi and 0 psi? Would the computer have changed that backbone angle to this region over here? Okay, and you can mess around with that, but it's just really important that you realize the starting point and the algorithm very much can change the outcome of your model. Okay, so that's a little bit more about protein structure, protein backbones, and if you're making your own peptides or modifying proteins, adding amino acids, it's pretty important that you consider these sorts of parameters, right? The backbone of your peptide chains.